We also possess areas that contain multisensory neurons capable of processing multiple senses. In these multisensory neurons, we see convergence of modality-specific information such as visual and auditory signals onto a singular multisensory neuron. And this convergence can lead to integration of these cues, which in turn improves our perception of the multisensory world. So this is just a little outline of what I'm going to discuss today. So I'm going to start with one of the behavioral outcomes of multisensory integration and go into the physiology, what part of the brain actually plays a major role in this behavior, the anatomy of this area, what's happening on the neuronal level, and how this ability develops. And then I'll move into some physiological results and impacts of removing one of the sensory modalities experienced. And then end with showing some behavioral results of how this impacts behavior. So I'll note just briefly that we have, and others have done, a numerous amount of behavioral studies looking at multisensory integration, different parts of the brains that have this ability, and how rearing animals in different conditions where we manipulate the sensory experience that they have, how this actually impacts the development and the physiology of this multisensory integration capability. What no one has shown is how this impact on the physiology actually maps onto the behavioral outcomes. And so that's what I'm going to show you today. So, while there are many different behavioral outcomes, benefits we get from multisensory integration in the brain, one that particularly pertains to what I'm going to discuss today is the enhancement and detection of localization accuracy. So in a localization task where a brief flash of light or a burst of sound is played across many different locations, we see that when these two cues are played together, there's an increase in the accuracy that the participants make. And this increase is significantly better than the best unisensory cue. So the area of the brain that plays a major role in this behavioral outcome is the superior colliculus. And I've outlined it in the human brain up here and in a cat's brain because we typically use cats in our studies. So the primary functions of the superior colliculus are detection, localization, and orientation of salient events. It has neurons, unisensory and multisensory neurons, that can process and respond to visual cues, auditory cues, somatosensory or touch, and nociception or pain stimuli. The neurons of these different sensory modalities are laid out in a map-like fashion in the superior colliculus, and they overlap and align with each other. And this can be seen here in this image here, where the visual cue from the rodent and the auditory cue being projected onto the cat, you see is activating the same area in the superior colliculus. So these maps overlap and align with each other. These sensory maps also overlay a motor map, which allows the function of the superior colliculus to be very efficient. But how are these neurons responding to these external cues? So if you were to take one multisensory neuron, we can measure the amount of impulses or firing that this neuron makes when you present a visual auditory cue. So if you have a visual receptive field here, shown in blue, an auditory receptive field shown in red. If you were to play a visual cue inside that receptive field, you will get a certain firing from this multisensory neuron. If you were to then play a burst of sound, an auditory cue inside that receptive field, you'd get a different response, from that multisensory neuron. However, we see that when you play both of these cues in the same receptive field at the same time, we get a response that is significantly greater than, again, the best unisensory response. And so we can say there's this multisensory enhancement happening from these cues integrating. Now, there have been a few principles that have been determined over many years of research that can tell us how the neuron is going to respond given an external cue. The first two principles deal with the spatial and temporal aspects of the cue. And so, simply meaning that spatial and temporally concordant cues shown here generally lead to a multisensory enhancement, integration and enhancement response. Conversely, a spatially disparate cue, where the visual cue is inside the receptive field and the auditory cue is played outside the receptive field, generally leads to depression in the multisensory response. And similarly, if you play these two cues at different time points in the same receptive field, the multisensory response will decrease, and so you see a decrease in the multisensory enhancement. The final principle just simply deals with the efficacy of these cues and simply says that if these modality specific, so the visual and auditory cue, evoke a strong response in the multisensory neuron alone, then the proportional enhancement you'll see is less than if they evoked a weak response. So if you compare the response here when they were strong, evoking a multisensory response versus here when they're relatively weak, the enhancement you see in the multisensory case is substantially greater. But now, while this is a very powerful feature of the brain, we see that at birth there are no functional multisensory neurons. So these neurons are not able to integrate these cues and get an enhancement of the response we see in adults. So here's just a timeline of a cat. And so when cats are born, their ear canals are closed and their eyelids are closed. So the only neurons that are responsive in the superior colliculus are those that respond to touch. At 1 week, we see that the ear canals start to open, so we see neurons that are able to respond to auditory cues emerge, and at 3 weeks, we see neurons responsive to visual cues. Now, during this multisensory convergence period, multisensory neurons are emerging. So we have neurons that are capable of processing multiple different senses. However, they're not able to integrate these cues together. So the response you typically see from these multisensory neurons are no better than the response to the best unisensory cue. And so multisensory integration capabilities are not seen in cats typically until about 4 weeks. Another important aspect that we see in development of the superior colliculus is that the receptive field sizes start out very large and then they contract as the animal matures. So on the left here, I'm showing you the receptive field sizes on the y-axis of all the three modalities. And so at the beginning, they start off very large, and then as the animal matures, they contract. And on the right here is an example of a multisensory neuron that is responsive to visual auditory cues. And so the gray area represents the receptive field. And so you can see that at 22 days postnatal versus 135 days postnatal, the size of those receptive fields are drastically different. And so the contraction in receptive fields improves the reliability of the spatial representation of the receptive fields and also increases that alignment across the sensory maps. All right. So what would happen, or what happens when we remove the sensory experience of one of the modalities, such as visual, by raising animals in a dark room so they receive no visual, non-visual interactions? How does this actually impact the physiology and development of multisensory integration? Well, one of the impacts we see is that the receptive field sizes are significantly larger. So on the top is a normal adult cat, and the black circle represents the visual receptive field, and the dark gray represents the auditory receptive field. And you can see in the dark reared adult animals, they receive no visual and non-visual interactions. The size of the receptive fields in both the visual and auditory case are significantly larger. The other important impact that we see is that they fail to develop those normal multisensory integrative capabilities. So whereas in normal animals, if we play a visual and auditory cue in the same receptive field at the same time, we see a response that is greater than the best unisensory response. In these dark-red adult animals, they're not able to integrate these cues, and so the response is no better than the best unisensory response. So it's almost as if they are stuck in this multisensory convergence developmental stage and are not able to progress to this multisensory integration stage. And so we have done a numerous amount, again, of studies rearing animals in different conditions where we show, we remove visual interactions, we place them in a room where there's a masking noise, so it's omnidirectional noise, they don't hear any auditory pattern input. We've raised them where they receive random signals, and basically from all of this research we have learned that the animals have to receive cross-modal inputs within the same receptive fields in order to develop this multisensory integrated capabilities. But what we haven't shown is how this impacts the behavioral outcomes. So how does the animals not being able to integrate these cues together to get this enhancement, how does this impact the behavioral outcomes? And so what I wanted to do was study this behavior in a detection and localization paradigm, specifically looking at the impact of dark green on the auditory enhancement of visual detection and localization. And so given the physiology that we know, I hypothesized that these animals will show no enhancement. And I'll go into how I measure this multisensory enhancement in behavior, because we can't simply do the same measurements that we did in physiology. So what did I do? So animals were born in a dark, light-type room. They received no visual, non-visual interactions until the testing period. This room was fitted with infrared lamps, so I used night vision goggles for everyday food, feeding, and care for these animals. They were trained at 1 to 2 years old, and the training and testing took place in complete darkness. So for the localization paradigm, I first trained these animals to fixate directly straight ahead. I then trained them to detect, localize, and approach a visual or flash of light and an auditory burst of sound shown across 7 different locations on this apparatus. So it's from -45 to 45 degrees. During training, the visual and auditory cues are played separately. So there'll be trials where there's visual-only cues being played, and trials where there are auditory-only cues being played. And I also interleave some catch trials where there will be no cue played, and the animal had to stay fixated directly ahead in order to receive a food reward. Once these animals have sufficiently learned this task, I then reduce the intensity of the visual and auditory cues to make the task much more difficult. So at the end of training before reduction, these animals are getting this right at about 75–80% or above at these different locations. But then after I reduce the cues, now they're only getting it accurate about 30% of the time. And for testing, I then added the cross-modal auditory visual cues. So now there are auditory-only trials, visual-only trials, visual and auditory trials, multisensory trials, and catch trials. And then I evaluated the behavior. So before I go into how I'm going to evaluate this behavior, I want to give you a little warning that it is going to get a little math heavy, so just bear with me. So in the physiology, we typically measure the multisensory response to the best unisensory response. But we can't do this in behavior to measure the multisensory enhancement. Instead, what we need to do is come up with a predicted behavioral response if there was no interaction between the cues. So the way that this model works is that for a given target location, let's say 0 degrees, I take all of the auditory responses the animal made when the target was 0 degrees for auditory-only trials. I take all of the responses the animal made when it was a target at 0 degrees when it was a visual-only trial, and I pull all of these responses together. I then randomly sample from that pool an auditory response and a visual response, and I compare these two responses together and pick the better. I then do this multitude of times where I'm just randomly picking auditory responses and visual responses and comparing them and picking the better. And this gives me the cross-modal predicted response given no interaction between the cues. And what we see by doing this is that the response that we see in this predicted response gives us a statistical facilitation effect where the predicted response is greater than the responses of the auditory and visual when they were played by themselves. So this is a null hypothesis model of predicting the behavioral outcomes if there was no integration or no interaction occurring between these cues. So what does the actual behavior look like? So this is data from 4 different animals, normally reared, so they have normal rearing and development. And on the left here, just to orient you, I have collapsed and averaged the data across the locations. And so here is an average auditory response shown in red, the average visual response shown in blue, and the average multisensory response shown in purple. This green dashed line is the statistical facilitation predictive response given no interaction. And so what we see is that in these normal animals, the multisensory response is significantly greater than the statistical facilitation. So because these animals are able to integrate these two cues, we see an enhancement in the response that's greater than if they were not able to integrate. And on the right here is just shown the responses across the locations, and just to show you that they are consistent across all locations. So I have two dark green animals to show you. So in the first dark green animal, and the graphs are set up the same way, you see that the multisensory response is significantly lower than the statistical facilitation prediction. And this is consistent across all the 7 locations. And in the second animal, we see that the multisensory response is no different than the statistical facilitation response. So because neither one of these animals actually beat statistical facilitation like we showed in the normal animals, we conclude that a lack of cross-modal experience precludes or blocks the development of multisensory integration in physiology, and thereby blocks the multisensory enhancement we see in the behavior. This is kind of like the last missing piece of the puzzle that I've added. However, you're probably asking yourself, why is there a difference between the multisensory response in the darker first animal, where it was significantly lower than statistical facilitation, versus the second animal, where it's no different than the statistical facilitation? And I have a hypothesis, but I won't be able to prove it. More animals will have to be tested. But these animals had a significantly different amount of training time. So if you have pets at home, or if you've ever dealt with other people's pets, you know that certain animals are smarter than others. And so that's the same in this case. Some animals take a longer time to learn this localization task. But because they received a significant different amount of training time, that means they received a different amount of sensory exposure. And we have shown in other studies that unisensory exposure leads to the contraction of those receptive fields. And so I'm hypothesizing that in this animal, it probably had some refinement in those receptive fields that allowed it to meet at least statistical facilitation, but not beat it. Whereas in this first animal, it didn't have any refinement or any contraction in the receptive fields, so that is why it was significantly lower. And I'll end my talk by discussing some of the human studies that some of our colleagues have done. No, they are not placing babies in the dark room and raising them that way. So in these studies, what they generally use are adult patients that were born with cataracts. So these people are born with cataracts, so they receive no visual pattern input for the first few months of life. And then they're treated for that cataracts, they have those removed, and they're allowed to grow up in a normal environment. And these studies look at how these patients' multisensory integration capabilities are different than normal patients. And what they found, just some of the results that they found, were that they performed significantly worse in an audiovisual speech perception task. So in these audiovisual speech tasks, typically you'll have an auditory-only trial where a word is being played with some varying in background noise. So you have a word played, they'll have either no background noise or a lot of background noise, which makes the word hard to hear. They'll have visual-only trials where it would just be someone saying the word, so it's like a lip reading. You have to read the lips to understand what they're saying. And then they'll play these two together. So you have the auditory sound in the lip reading, and generally we see an enhancement in the response when these two cues are played. However, in these congenital cataracts patients, they don't get that bump when the two cues are played together. So this is possibly due to either lack of multisensory integration or an impaired multisensory integration ability. The other result that they find is that they experience a reduced McGurk effect. And so the McGurk effect is simply when an auditory sound is played with a visual incongruent lip movement. So if the sound ba is played with the lip movement da. Typically, normal people get a new perception and they either hear a new word or they hear some type of fusion of these two words. Whereas in these congenital cataracts patients, they get a reduced effect of that. So again, possibly and probably due to the lack of multisensory integration or impaired multisensory integration ability. So with that, I would like to thank my two PIs, Dr. Barry Stein and Dr. Rowland, my other committee members, Dr. O'Hearn and Dr. Stanford, my lab, so Alex Nakos, Wade, Nancy, and a special thank you to Naomi Bean for teaching me how to do all the behavioral experiments with the animals. And last but not least, my family for showing and being fully supportive. So with that, I'll take your questions.
So we have questions?
So in the timeline for the development of multisensory integration and studies of physiology, how does this relate to the development or lack thereof of multisensory integration capabilities in cataract patients? I mean, do they ever show this capability with experience later on?
Well, no, because the experiments that they're typically doing are in adults, so they would have had a normal amount of multisensory experience from just normal environment. But even as adults, you're talking about 14, 15, 20 years of experience of this does not allow them to develop this multisensory integration capability.
So does that fit well with experimental studies where you can control the environment as in animal studies?
Yes. So, so we have done experiments where we, again, rear the animals in the dark, and then we will place these animals in a normal environment to see if they're able to develop this multisensory integration capability. And we see that even after a couple of years of them in this normal environment, they do not develop it unless we present these two cues together in space and time.
So what's different between the training environment and the normal environment?
So in the training environment, we are allowed to basically show auditory visual cues in the same receptive field and glassing them with this. Whereas in a normal environment, there's a lot of varying in how these auditory visual cues are actually coming into the humans. And so by controlling this variability, we're able to quickly develop this multisensory integration capabilities. Yes.
Yeah, so we didn't ask questions on the committee, but here we ask questions.
We'll have a separate.
Oh, what was that?
But you can ask questions.
Yeah, still, yeah. Yeah, well, as far as humans go, I was wondering whether they show normal unisensory functioning. And I take it that human samples were matched for like birth, gestation age and all that stuff because.
Yes, and they even match them for lip reading ability because these patients do show deficit in lip reading, and not all normally reared people are able to do lip reading as well as others. And so even when they match the lip reading abilities to humans that have that same comparable ability to do lip reading, they show reduced interaction and McGurk effect and perception.
And do they have enough variability in the amount of time that it took to get the cataracts out? And do you have enough variability in the animal studies you've done to see whether the amount of multisensory impairment relates at all to the amount of time that they were dark reared? I mean,
Sorry.
humans were dark reared.
Yes,
But,
say that again. Sorry.
So some people got their cataracts out quickly and probably some it took them quite a long time. And I thought I remembered from old studies that the amount of time that they had the cataracts in was reflected in their performance on
Yeah, so
and stuff like that.
yes. So if you were to So in the studies that I've mentioned, they usually go from about the average was about 8 months before these cataracts were removed in the data that I presented. But if you were to remove these cataracts during that important developmental stage, they would still be able to develop this multisensory integration period ability.
Well, you think they develop it normally? You think they develop it somewhere in between, or?
I don't know, but I would
You don't know?
hypothesize that it would develop normally as long as they were in the critical developmental period.
So what would you call the critical developmental period for the cats and for the humans? Like, you can just guess.
So so I don't know for humans, but for cats, this multisensory convergence during these first, couple of weeks are the critical.
So you think if they had dark period for 2 weeks and then not dark period, they would be fine? They would be typical.
We're taking a guess here. I'm hypothesizing that they would be able to develop this normally. Now, I will add that we have shown that the adult cats are, the brains of the adult cats are very plastic, and we can recover this multisensory integration ability. But again, that's through intense training or blasting them with crossbow experience versus just them being able to just walk around the environment.
Right. Well, for humans, a lot of the cross-modal stuff would be social interaction, probably. Probably, I don't know if it would be the same for cats or not. So, in that way, you kind of are getting an intense cross-modal experience. When you deal with people, you're always dealing with audio and visual stimuli, pretty much. At least during an active interaction.
Yes, but so in a normal environment where we're hearing speeches and we have all these different light and visual and auditory cues coming, they're not necessarily coming, how do I say this? We're not actually controlling that they're coming from the same space and time. There's a lot of difference in angles that they're hitting the brain and auditory signals. And so there's a lot of variability versus if we can control that these two cues are being played at the same space and time, then I believe that is necessary for this ability to develop. And that's why it's not developing normal.
So the hypothesis that is the amount of training with the unisensory input that showed differences between the subjects.
So say again?
So you have a hypothesis that it was the amount of unisensory training that led to a difference in your subjects? Is that right?
Yes.
So
multisensory response.
yeah, right. So do you think that given many trials, you'll continue to see some kind of improvement, or are there just
No, so we did an experiment where we took these darker animals and showed random, just unisensory exposure to these animals, and these animals did not develop the multisensory brain capabilities. It was only after they experienced cross-modal cues that they were able to develop this multisensory brain capabilities. So we tested that.
So let me give you another postulate with regard to developing to reach statistical facilitation. As you bring the animal up and you train them, and you give them unisensory experience, you can't help but the animal getting cross-modal experience at the same time. So the duration of training is much longer in the second animal than in the first animal. So the amount of cross-modal experience the animal gets in that environment is also much greater than the first animal. And so that may have been the reason why that animal reached statistical facilitation, and would also be more consistent with the idea that cross-modal experience leads to development of sensory information capabilities physiologically and then behaviorally. One last comment about the last piece of the puzzle. One of the interesting things that you're looking at when you look at the behavior is the multisensory function of the entire brain. Because before we were looking at physiology using the colliculus as a model, we only showing in fact that the same principle applies when you look just at the blood flows and you look at the behavior, may tell you the rest of the brain is following what that model structure is doing. It's just an aside. So if there are no other questions, you're on to your exam.